Good day everyone. My topic is about the preparation and purification of colloidal system. The formation of colloidal particles involves either the dispersion of large matter or the condensation of small molecules or ions. The first method is by dispersion. Dispersion is the method of breaking down of big particles by grinding in a colloid or by ultrasonics. Dispersion leads to extensive subdivision that causes small particles to reunite. Due to the mechanical forces and due to the attractive forces between the particles. Also, it can be obtained by adding an unreactive substance to reduce the chances of the particles uniting with another during grinding or by wet milling, in the presence of surface active material. An example is the grinding of a mixture of sulfur and glucose, dispersing the resulting powder in water. For example, it is a milk powder and water. So, as you can see, it has dispersed. The milk powder dispersed in the water. So, this is also an example of dispersion. And also, another example is dissolved glucose from the salt by dialysis. So, another method is by adding an emulsifying agent, such as soap in an oil-water solution to produce an emulsion, or an egg yolk in mayonnaise made of oil, and calamansi juice. This is calamansi and this is water. So, we have calamansi. So, when we put calamansi in water, it has produced emulsion. So, the calamansi here is the emulsifying agent and the water is the liquid. So, when we mix it, as we can see that the calamansi is the emulsifying agent. This is also involved in dispersion. So, calamansi juice is an example of dispersion. Another is vinegar. Vinegar is an example of emulsified solution. In addition, peptizing agent reduces big particle to colloidal size. Enzymes that break down food particles during the digestive process are example of peptizing agents. Also, homogenizers are machines that emulsify or blend a substance. Milk is homogenized to prevent the fat from rising to the surface. So, when companies or when milk companies are producing liquid milks such as Alpine, Angel Condensado, or any kind of milk, they're using a homogenizer to homogenize that milk so that it will prevent the fat from rising to the surface. If we have dispersion, we also have condensation. Condensation method is the process of combining molecules in supersaturated solutions to precipitate or coagulate them to colloidal size. Substituting a poor solvent for a good one, cooling and various reactions such as hydrolysis, oxidation, or reduction can be used in condensation. Dialysis is similar to the process of osmosis. Both are used to remove excess ions from colloidal mixtures. When a membrane allows the passage of water and small particles, as the cell membrane in your body. That cell membrane is an example in the cell membrane in our body, because that cell membrane allows only non-toxic substances to enter our cells and prevent toxic substances to enter from our cells. That process is called dialysis. The membrane is called dialyzing membrane. It does not permit much larger molecule to pass through. I have a video. I will show you a video that is about dialysis.
Your doctor may recommend dialysis, a procedure that removes waste products from your blood if your kidneys are not functioning properly. Healthy kidneys filter out waste, excess fluid, and electrolytes like potassium and sodium from the blood. These waste products exit the body through the urethra in the form of urine. If you have kidney disease, your kidneys gradually lose function, leading to an accumulation of harmful wastes and excess fluid in your body, increased blood pressure, and bone or blood problems. Dialysis can also be done to treat acute kidney failure, which occurs when your kidneys stop working suddenly, or to remove drugs or poisons from your body. In one type of dialysis, called hemodialysis, your doctor will use a machine called a dialyzer to clean your blood outside your body. Some weeks or months before you begin hemodialysis, your doctor will prepare a vascular access site on your body. Your doctor will create either an arteriovenous graft or an arteriovenous fistula to access your bloodstream. To begin, he or she will numb the area with local anesthesia. To construct an arteriovenous graft, your surgeon will make a small incision in your forearm and insert a soft plastic tube called a graft to connect an artery to a vein. To create an arteriovenous fistula, your surgeon will make a small incision in your skin and connect an artery with a vein to make a larger vessel called a fistula. Once your vascular access site has healed, your doctor can start hemodialysis. To begin, your dialysis nurse will insert 2 needles into your AV fistula or graft. Once the circuit is set up, your blood will flow slowly out of your body and through the dialyzer. Inside the dialyzer, your blood will encounter filtering fibers and an absorbent cleaning solution called dialysate. The fibers will remove waste and excess fluids and electrolytes from your blood. Your cleaned blood will emerge from the dialyzer and continue through the tubing back into your body through the second needle at your access site. When your hemodialysis session is complete, the needles and tubing are removed, and you can go home. In another type of dialysis, your doctor will use the membranes lining your abdomen, called the peritoneum, to clean your blood without removing it from your body. Before you begin peritoneal dialysis, you will have an operation to implant a soft tube called a catheter. Your surgeon will insert the catheter near your belly button, creating a port through which the dialysate will enter and leave your abdomen. Each peritoneal dialysis session is called an exchange. First, you will use your catheter to fill your abdomen with dialysate for a period of time determined by your doctor. Through the peritoneal membrane, waste products and extra fluid are continuously drawn out of your blood into the dialysate. Along with the waste materials it absorbed, the dialysate will drain out of your abdomen into a disposable collection bag during your exchange.
We saw in the video the process of dialysis. We know that there are 2 kinds of dialysis. So, I will summarize. A human kidney dialyzes blood to remove waste products produced in metabolism. In certain ailments, the kidneys fail to function and waste products and toxins will accumulate in the blood. This can cause uremia poisoning, which can be fatal. An artificial kidney machine uses dialyzing membranes to help remove the smaller waste materials while letting the blood retain its larger protein molecule. The process is called hemodialysis. That's all. Remember, the ways in preparation and purification are dispersion and condensation. Powdered mixed-in liquid is an example of dispersion, and oxidation and reductions are example of condensation. Hope you learned a lot.